All right, so what we're going to be talking about today is how to develop and write a review paper. And we're going to be treating this sort of in the broad sense. So really talking about all kinds of review papers. And then I am also going to focus some of the time in the second half of it on meta-analysis in particular. All right, so hold on, there's still people coming. I'm trying to let in. Okay. All right, these I think are the bookkeeping. Most of these I've already shared with you, the links that are in the chat. Please do mute your microphone just because I think that makes it easier for us all to hear. All of these are things in the chat, so please do check that out. I have links to the survey from Botany360. I have some links about review submissions to AJB and also Botanical Society of America webpage. All right, so, I wanted to start out with saying that writing reviews is super fun, and it's also a really important tool for moving a field forward. It's a great way for you to become an expert on a particular topic, and I think it can have really great consequences for your career development, because reviews, if they're done well, are often relatively well cited, and they can really help increase your visibility in a particular field. So I think reviews are really great. And I also think it's really important to remember that anybody can write a robust review. I think we usually consider that people who are already well-established experts in the field are the only ones that can write good reviews. But I definitely think that that's not the case. I think there are real advantages and disadvantages depending on your level of experience with a topic. So, of course, people who have a lot of experience on a topic have all of that familiarity and expertise, and that can contribute in really important ways for how they develop and write a review. The disadvantage of having all that experience, particularly if a lot of it stems from your own lab's research, is that those folks tend to be relatively biased on the field because their perspective is driven so much by their own personal research. And if the goal of a review is to describe general patterns, look for sources of heterogeneity, things like this, then that bias that stems from our experience can be a real disadvantage. On the other hand, people who are relatively new to a topic, maybe because they're early career or just because they're moving into a new area, they have the disadvantage of not knowing the topic very well. So they may miss out on some of the parts of the literature that are important, or they may not know exactly what the best questions are to address in a review. On the other hand, they tend to be more objective, so they have less bias that they bring to it. And in that way, approaching a review as a relative newcomer to a field can be a real advantage. So my point here is just that everybody, depending on whatever kind of experience they have, can write a good review. There are different disadvantages and advantages depending on your previous experience. And our goal here is to really demystify the process of how we should approach writing reviews, and in particular to highlight the value of taking systematic review approaches, because those help us overcome both bias and a lack of familiarity with a field. And so I think for everybody, using systematic review methods results in the most robust kind of review, and therefore helps move the field forward in the best way. So that's what we're really going to be focusing on, is how to do systematic review methods. And then whether or not you combine that with meta-analysis, it depends on what your question is. And we'll finish up by talking about how to do meta-analysis in particular. So that's our goal here. Here are the different steps of what I would say is the most robust approach to developing and writing a review. We're going to go through all of these steps. And actually quite a lot of it comes before you even start assessing the literature. So setting yourself up with a review is really important, and it should take all the same kind of care and attention that you put into designing any kind of scientific study. Writing a review is no different. If you approach it haphazardly, then you're probably going to end up with less than robust review. You would usually not do that in other scientific pursuits. And I think it's the same for writing a review. All right, so here we go. First of all, I just want to introduce myself a little bit so you can see how I'm approaching this and what my expertise is in this area. So I'm a plant functional ecologist. I'm at the University of Hawaii at Manoa, where I'm in the School of Life Sciences. I've been on the faculty here for the past 13 years. And my research examines mainly two main questions. One is, how does plant functional strategies and stress tolerance change as plants grow up across ontogeny? And then also, are island plants fundamentally different from continental plants? So this takes a lot of experimental approaches, and we also do quite a lot of research synthesis. So I just want to point out here that I am not an innovator in research synthesis. That's not my training, or that's a small subset of my training. I'm really an ecologist. And so I use research synthesis in my own research to try to understand sort of general patterns and why patterns differ across different sources of heterogeneity. But I rely on the people whose real expertise is in research synthesis to provide me with ideas of best approaches, skills, things like this. Like I said, I do a lot of research synthesis myself. This is one of my favorite kinds of science. And I've published 7 integrative reviews. So those are narrative reviews, 4 meta-analyses, and I currently have 3 new reviews in review. So this is definitely something that I love to do. I think it's really exciting part of science, and I hope that I can share that with you today. All right, so I do have some bit of training in research synthesis for my postdoc. I spent it in the UK, collaborating with Yulia Kuricheva, who's really an expert in meta-analysis in ecology and evolution. And the great thing about having a postdoc that was dedicated to research synthesis is that I had the room and the time to really develop my own sort of expertise in this. And I spent my whole first year really reading extensively in the field of research synthesis. And largely this was at the time in sort of medical and sociological fields, because they innovated meta-analysis and have used it extensively for much longer than we have in ecology and evolution. And I was able to learn a lot about these approaches from those fields, and then it's easily transferable to ecology and evolution. And I think that really helped me develop sort of good practices early, which I now see it becoming, everybody's doing it. It's totally widespread. But also the point is that research synthesis is itself its own field. This is a formal scientific method. And so if we take the time to learn about how to use this method effectively, then I think we can do much more successful research synthesis, and we can achieve the kind of generalizations or generalizability that we're really hoping to do when we write review papers. So because this is its own field, there's a ton of literature out there. And it's great. So much of it now is in ecology and evolution. I find this all to be super helpful. So this is a great book that came out several years ago now by Yulia Kuricheva, Jessica Gurevich, and Carrie Mengerson. And this is an edited volume, so it includes chapters by lots of folks. And it's a really great resource for if you're doing meta-analysis in particular. I highly recommend this. But you can also find lots of great literature just in journals. There are many journals and even ecology and evolution journals now that publish on systematic reviews and meta-analysis. And I included in the chat a link to a list of references that I compiled, which is just a very partial subset of the literature that's out there. But those are the papers that I have found most helpful in the past several years. So I'm sharing them with you, but this is just meant to be sort of a primer. If you're really excited about this and you want to learn more, there's lots of ways for you to access this information. Okay, and then finally, I do have some editorial experience, and in particularly with reviews. So at American Journal of Botany, I'm the reviews editor. And so I get the wonderful job of inviting reviews and handling review proposals. And so I'm really thinking a lot these days about how to attract more reviews, what kind of reviews are most helpful and reach the largest audience, or in some cases reaches just the right smaller audience. and especially excited to try to promote reviews and meta-analysis to early career folks as a way of boosting their visibility and expertise. And so I'm happy to talk to you about that if you're interested. All right, so let's get started. You think you want to write a review. How do you approach this? I think the main point to start us off here is that I propose you start this with a lot of intentionality. So sometimes we just sort of wander into a review and then haphazardly go about it. I'm going to argue that that's not the most effective approach. And if you find yourself doing that, that's okay. Just then shift gears a bit and do these more formal steps so that you can achieve the kind of consistency and rigor that we're looking for in reviews. So let's get started here. These are the steps I think are important for systematic reviews and how you get started. So these are, I'll just walk you through what these mean. So to start, there are many different ways that we refer to reviews. So how do you know which one you want to actually do, right? So they're called, this is what I hear them called most commonly, at least, a review, a narrative review, an integrative review, a systematic review, a meta-analysis, a perspective, an opinion, or a synthesis. And unfortunately, I think you could talk to different people and they would, in large part, have variable answers as to what these are or what the differences are. I'm not sure it's worth our getting into the semantics of this, but it's definitely these words are out there. And I would say that the two important things that differentiate whatever you call a review are these two things. First of all, are you compiling your literature in a systematic way or not? That's one important distinguishing characteristic. And the other is, how are you assessing the evidence once you get that literature compiled? Are you assessing it qualitatively or quantitatively? And then I could try to put these different types of reviews into these different categories, right? So here's non-systematic searches versus systematic searches, qualitative assessment and quantitative assessment. Oh, somebody needs to mute themselves. Can you please? Thanks. Okay. So a qualitative non-systematic review, it would be like a perspective or an opinion. A qualitative assessment of a systematic review. So these are when we compile the literature systematically, but then we do like a narrative review, an integrative review, a systematic review, a synthesis. If we're going to analyze the literature that we compile statistically, then we end up with a meta-analysis. And I would argue that if you do a meta-analysis, that you are sort of required or expected to do a systematic search in order to gather the evidence that you're going to analyze. And I would say that if you do a meta-analysis using non-systematic search approaches, that you end up with a flawed meta-analysis. So don't do this, right? All right, here's the thing that's going to be that you need to keep in mind when you're deciding these different kinds of reviews is to what degree are they going to be biased? So when you are writing a review, and the sort of conclusions that you're drawing or the general patterns that you mean to characterize, we always, I think, need to be mindful of the potential sources of bias in our review, because that could then mean that our review is either robust or not robust or constrained in how far we can generalize, et cetera. So this is the main thing that will lead to a poor review, or that we at least need to be mindful of its role. And I would say that a flawed meta-analysis, we don't want to do that. Perspective and opinion papers are purposefully biased, right? That's the whole point of them, is that the author has some point that they're trying to argue. So an approach that's flawed or some new opportunity. So these are meant to be biased. That's why they're perspective and opinion papers. So we know when we hear these words that this is going to be a biased take on something, but that's okay. That's the point of it. For these two kinds, the systematic search helps us minimize bias, and the meta-analysis tends to have the least bias because the evidence is assessed statistically rather than just through our own interpretation. But I would say that there's an important role for all of these different kinds of reviews, except for flawed meta-analysis. All right, so I mentioned how bias is so important. What do I mean by that? Whoops, what's happening here? So what kinds of bias can happen in a review paper, and how can we overcome this? So probably the most common source of bias is that the literature that's assessed by the review is only a subset of the literature on that topic that's out there. So excluding relevant literature is necessarily going to lead to a bias if we compile that literature in some particular way. So this usually arises because of poor search methods. So you're missing some of the search terms, or the search systems you're using aren't comprehensive, and I think this is really common, there's mission creep. So you start out thinking that you're reviewing this particular topic or question, and the more you read, you sort of change your idea of what the review is, and now it's on something slightly different. And I think that's probably typical and not a problem, except that if you haven't then done some searches to identify the literature related to your new focus, now you've introduced bias to the literature that you're reviewing. So excluding relevant literature is probably really common, but something that we want to avoid if we're trying to seek general patterns. Sometimes we exclude relevant literature intentionally, and this is usually associated with somebody's a priori ideas or assumptions about a topic. So they may be prioritization of certain scientists or their own work. There may be a bias in terms of they want to focus on particular study systems or particular kinds of data. Sometimes this is fine as long as there's transparency, right? So as long as we say this is what we're focusing on, it's a subset of what's out there, then that's totally fine. If you aren't transparent about that, then the reader has no idea why some of the literature is being omitted. And I think here's where including your own unpublished data can become really problematic, because if you want to include unpublished data in a general way, you need to seek out all unpublished data. If you're using only unpublished data from your own lab or of your friends, now you've intentionally excluded other data, and that introduces bias to your review. So combining a review of published and unpublished data needs to be handled very carefully. Obviously, language barriers can introduce major biases to reviews, and these can be associated with biogeographic biases. So this is something that we all need to be aware of. I'd say it's extremely common in reviews, and often we can't overcome this, but we need to be aware of it so we can acknowledge it as a legitimate source of bias in our reviews. Publication bias can be a problem, in particular if for a given field there's a propensity to not publish null or negative results. Then that can lead to an overestimation of the commonality or the extent of an effect, because only the positive and significant studies are getting published. So publication bias is a huge issue. People are well aware of this, and there are ways to try to address or investigate the likelihood that this is affecting your data sets. Some people also argue that we should not rely only on published data, but you should seek out unpublished data that's available in, say, gray literature, agency reports, dissertations, things like that. And there are recommendations for how to go about that in a systematic way. So in general, the point of bias is just knowing that it's there so that we can try to overcome it or acknowledge it or account for it, right? So we temper our conclusions in light of these biases. The worst thing is having a bias in your review that you are unaware of, because then you try to generalize and you're constrained by your own data set and you don't realize it. So we want to be aware of this so we can address it. And as much as possible, we want to minimize it. And that's where systematic review methods and meta-analysis can be really helpful. All right, so here I am pointing out that meta-analysis is the least biased. These are more prone to bias because they rely on our own interpretation of the evidence, and we ourselves always carry implicit biases. Nonetheless, these are super common, these integrative, systematic, and synthesis reviews. I think they're really important. I'm not arguing that we get rid of them, but I think we need to acknowledge the potential sources of bias that go into them. All right, so now let's get into some of the mechanics of how to actually do this, right? So you may have an idea now that of what kind of review you want to do. How do you get started? And I would say at this point, here's where you're approaching this like you would any other kind of scientific method, right? So we want to be really careful about this, intentional and detail oriented. So I would suggest you get a lab notebook like you would for any other experiment or study, and you take really detailed notes of every decision you make in this process so that you can document this as an official protocol and publish it with your review. And that leads to two things. One, there's greater consistency in how you assess the evidence that you are compiling. And two, it provides transparency to the reader. So the reader now has a sense of what you did or did not do in the decisions you made in compiling a certain body of literature to review. And that really helps the reader understand what the review relates to and any potential limitations. All right, so how do you identify your question? If you're already an expert in the field, you probably have a good idea of what the question is you want to address. If you're relatively new to a field, this may be a tricky thing and an important and difficult first step. I think the first thing to do is decide what is the goal of your review? What is its job, right? So there's different types of reviews based on what we're trying to do. Sometimes you're just trying to compile all the information on a topic or place or a species into one important reference. I would call that a summary review. It's not synthetic, it's more like a compilation of information. That can be super useful, especially I think it has pretty good longevity because it may reach a relatively narrow audience initially, but it's probably going to be valid and reliable for a long time. So these summary reviews are, I think, a little less exciting, but can be really important resources. So that may be your goal. On the other hand, maybe you are an expert and you think, or a new person to a field, and you think people are approaching a question or a topic wrong, or that you want to argue for a new perspective or a new approach. This is sounding like a biased perspective, right? This is more of like a perspective or an opinion paper. Also really important, different kind of role. And I would say not necessary to use systematic review methods because you're arguing a certain point. I would say a synthesis is, to me, the most exciting kind of review. Here's where you're trying to pull together different bodies of work. So this could mean different approaches to investigating a topic by different scientists who maybe don't talk that much, so they don't realize how their different perspectives may come together. You could be synthesizing across biomes, across different kinds of traits, different interactions, whatever. This synthesis is meant to provide new insights. And I think those are exciting reviews and really can change the direction that a field is taking. When meta-analysis becomes particularly useful is when there's a particular question, so a hypothesis or theory that somebody has proposed that has now a fairly large body of literature that has tested that question, but the evidence seems mixed. So you see a lot of this in the literature. Sometimes people have seen this and sometimes people have reported this. And it seems like there's no consensus. That's when meta-analysis is really helpful because meta-analysis provides us with statistical tools to test for sources of heterogeneity. So if there's total consensus in a field, then a meta-analysis is not that helpful because it's just going to tell us what we already know. It's really helpful when we don't know why sometimes it's yes and sometimes it's no. And the meta-analysis can help us identify that statistically. So we're not having to rely on our own subjective interpretation. We can actually do it quantitatively. And then probably most reviews have a job of identifying data gaps in future directions. That's always hopefully part of a review, is emerging areas that we still don't understand well. And that's a really important outcome of review papers. So the question you ask depends on what your goal is, which of these things you're trying to do with your review, and then of course your particular topic. So the next thing you want to do, diving into the topic, is decide on the scope of your review. So what is the body of research that you want to synthesize, right? So this is a tricky, I'd say this is a tricky step because there's a real balance here. If your scope is too large, then your review could take you like your whole life, and it could be too long, and it could be too messy. If it's too narrow, then it may not have the size of literature that you want for a review, and it may reach only a small number of people. So finding this balance that's rigorous and providing new insights can be tricky, and of course depends on your field and topic that you want to investigate. But I recommend that you take a lot of time at the beginning of your review setting the scope, because this is going to determine the literature that you're going to compile. And here's where that mission creep and the haphazard approach can be problematic, because if you're changing scope as you go, then you're probably not capturing all the relevant literature. And you may be not capturing it in a biased way. So the type, and this is relying on my work, so this probably you have different ideas here, but I would say the type of scope that you might consider is like, what's my biogeographical scope? What kinds of study approaches am I trying to synthesize? Is it experimental? Is it genomic? What am I talking about here? What are the outcome drivers? So these are the sort of treatments or factors that you think may be driving the response of interest. Here's the response, the outcome. So basically, what kind of data are you looking for? Are you looking for trait expression? Are you looking for fitness, population dynamic parameters? What type of data are you trying to find? And are those data available? And all of this takes a lot of care in the beginning. And I'm just pointing out here at the bottom that you've probably heard of PICO criteria, and this corresponds to that quite easily, right? The populations, interventions, comparators, and outcomes approach, which stems from medical research, but I see it quite a lot in environmental synthesis now. Identifying these things before you start can really help you be careful about the literature that you're compiling. Now, defining the scope is so hard if you don't know the literature well to begin with. So this is the step that's probably the most tricky if you're relatively new to a field. And I would say the only way to do it is to start reading, right? The more you read, the more you get a sense of what are the extent of these factors in terms of how much diversity there is in the literature and how much you think they're going to matter to the outcome of the question that you're trying to review. So this step probably takes a while, and it benefits tremendously from becoming familiar with the literature before you start formally assessing it. If you're already an expert, then setting the scope is probably really easy. And so also, if you're relatively new, talking with experts in the field might help you settle on a scope quicker as well. Part of the scope is going to come back to data availability, because you need to have the data in order to do an assessment or a review. So your scope can be so big if there's tons of data, or it can be more narrow, but then you run the risk of there not being enough data. So that's where data availability is important. And I would say that in general, published or archived data is very easily accessible. So that's always included in reviews. Published data in the form of theses or dissertations are commonly excluded, even though they're probably widely available, but people don't always think to include them. And sometimes it's tricky to know if a thesis hasn't been published or not. If it's been published, then you might be double dipping here. So you have to be careful of that. If you're thinking of including unpublished data, that may be especially relevant for some questions about, say, management-related topics or conservation. Agencies may have a ton of data. Whether or not you can access that is something that's beyond the scope of what we can say. Maybe it's available, maybe it's not. You should not be getting unpublished data of your friends because that's biased, right? If you want to include unpublished data in general, the appropriate thing to do would to be putting out a call for any unpublished data on this particular question. And that way, hopefully, you're capturing the unpublished data of people you don't know, and it will be less biased. And then finally, a really important point is that sometimes the data are not available and they shouldn't be compiled and then analyzed and re-released through a review or a meta-analysis. And this is especially the case for Indigenous and cultural knowledge. I think there are important data sovereignty issues here, and people need to be really careful to think about whether, even if they can access that data, if they really have the right to use it and distribute it. And I would say that this is a really important issue and something that you need to be really careful about if you're dealing with biocultural data in your review. Defining the scope and identifying the question is usually more or less an iterative process, right? So these two things are going to go back and forth as you become more knowledgeable in the field. You may then refocus your question, and that may then help you define the scope and so on. So I'm putting this in a linear format here, but I'm noting that these two steps in the beginning could take a long time, and they may go back and forth and back and forth. And taking lots of notes helps you create a record of your decision-making and how you're getting to this scope. And hopefully in the end, you'll end up with your sort of clear framework of the scope that you mean to review to address the question of interest. Before you actually go on and do any searching, I think it's important to, at this stage, think about who your audience is. And the reason that this is important is because for some kinds of topics, having those audience members as part of the authorship team will totally determine the success of the review or not. So in particular, I think for management-oriented reviews, biocultural reviews, having members of those communities as co-authors is essential. And I'm also noting here that you might consider looking at the target journals that you think you might publish this in, so the journal in your field, and that you know publishes reviews, and see if they have any field-specific requirements or expectations. Some fields these days have pretty strict requirements for publication of reviews that include all of these initial steps in really formal ways. And you want to know that before you've done the whole review and have to backtrack, because that would be really painful. So that's why I'm proposing that you do this early, so that you're careful enough to be following all those field-specific expectations from the beginning. And if you're planning a review that has a particular audience that you think about having a co-author, if you don't yourself have that experience or expertise, to help make sure that you're doing this in a respectful way and also an authentic way, right? That you're actually reviewing the evidence that you mean to review. Okay, at that point, then you're ready to develop the formal framework of your review, and we usually call that the inclusion criteria. So the inclusion criteria are basically like the formal list of what studies need to meet in order to be assessed as part of the review. So this is usually published with the review. It's often referred to as inclusion criteria, but even if it's not, you can say it in a more casual way. The point is that here's where you're telling the reader what literature you're including in your review and which literature you're specifically excluding. And that then provides full transparency of the scope of your review. So the reader isn't left thinking like, well, why didn't they include this kind of biome? Or how come they didn't include those sorts of traits which are clearly related? The transparency helps set the stage so that we understand where we can generalize the results to. So these, you can see, these are all the same things that I discussed with scope, right? Here we are just formalizing our decisions about these different parts of scope. So this is what comes about from doing all that reading. So you decide like, okay, there's a ton of work on this in some system and not others, so I'm going to focus on the one that has all the evidence. I'm again including things that I know from my own work are the kinds of inclusion criteria that I look for. So this would be specific to your particular field, but these could be things like the biogeographic scope, what kinds of plants, taxonomic or growth form you mean to review, what kinds of studies are you including, experimental only, only in the field, say only studies that have done intraspecific or congeneric comparisons. Whatever it is you're trying to get at, you need to be clear about what kinds of studies you're including. Sometimes there are specific requirements of those studies to meet your criteria. So for example, they must have control groups. They must be grown from seed if you're looking at seedling dynamics. They must be free of symptoms of disease or herbivory, which may then interact with whatever factor you're trying to focus on, whatever it is. I think this is a really important point. I always include as an inclusion criteria that the studies have a robust study design. And this is my way of excluding studies that I think aren't very good. And that's important. A review doesn't have to review and include every single study on a topic. If you really do think some subset of studies have taken some kind of flawed approach, you can exclude those, right? The point is just that we're being transparent about it. So for me, I always say that they have to meet basic study design expectations, like no pseudo-replication, no confounding variables. They must report intraspecific trait variation. And that helps me then flag the studies that I don't think should be included in the assessment. What kind of outcome variables are you interested in? So what kind of data are you actually trying to synthesize? Is it fitness measures? Is it pollination services? Is it functional traits? I don't know, whatever for your particular study. And then the last one, this is really most relevant if you're doing a meta-analysis, data availability, right? You can't include a study in a meta-analysis if you can't access the data, because meta-analysis is quantitative. So those data have to be available either in raw form, like archive data. That's actually the most useful. That's super easy to handle. But it could also be reported in tables or even in graphs. And we can extract data from graphs using digital software. So that's okay. But for meta-analysis, they need to include things like sample size and variance, because that's the key thing about meta-analysis. It's not just comparing the magnitude, say, of difference between groups, but that within study variance is a weighting factor. So we have to have sample size and variance, and those are often, unfortunately, not included in archive data for studies that include lots of species. That's an often deal breaker for me when I do meta-analysis. Now, here's an important point. If the data are not available in the published form, you have the option of deciding whether you want to contact authors to request unpublished data. In my early meta-analyses, I did that, but I don't anymore, because now it's more common for people to publish their data. And also, I find that the success rate of contacting authors is super low. So it's just not worth the time. But it sort of depends on what your question is. If you think a lot of the data are unpublished or unavailable, it may be worth your time to contact those authors and request the data. And I've had my own data requested many times, so I think this is pretty standard. And people aren't, I don't think, surprised when they get these emails. They're just annoyed and maybe don't answer. Okay, once we have those inclusion criteria set, and we should do this early, then all of this planning process can be formalized in a review protocol. So the review protocol is like the methods section of your review. And actually most even integrative reviews now do expect you to report some of these details or all of these details. So I would say that after all this planning, which does take a long time, but it's going to set you up for a really nice review, you want to sort of synthesize what you've done at this point with an official protocol. Up till now, you probably have loads of notes, and here you are making an official protocol, which of course you can edit and revise as you go, but is a good starting point and can really confirm your thinking as you move into the next steps of compiling the body of literature that meet your inclusion criteria. All right, that's a lot of information because there's a lot that goes into planning a review. I really do think this is important to treat it the way you would any other study, with all this kind of intentional planning. But I have a break here in case you need a break, and also to answer any questions that may have come up. Yeah, so Yinfan, thank you. So I think what's probably typical is that you've done all this work, you have your inclusion criteria, then you start going through the formal literature compilation and assessment. And as you go, you discover some new inclusion criteria or new keywords or sort of body of literature you missed that you didn't catch initially. It's always fine to include that as you go. The problem is if you're following systematic review, you need to then do systematic searches at that point, right? Or if you have a new inclusion criteria that you're instigating after you've already assessed, say, 100 papers, you have to mark that so you can go back to the first 100 and make sure you were consistent to address those new inclusion criteria to the ones that you didn't include that for. So this is the thing with the haphazard as you go, as you're changing your plan the whole time, you're sort of like being inconsistent with whether or not a paper meets the criteria that you want, or whether there's potential papers that are relevant that you've missed. So as much as you can early be confident about your inclusion criteria, that's really helpful. And then as you go, just be really careful that you're taking notes and backtracking and filling in so that you're not sort of becoming inconsistent or missing things as you go. It's a really painful thing to change your inclusion criteria when you're almost done and have to backtrack and double check all those studies to make sure that none of them needed to now be excluded, right? Yeah. Yeah, I mean, I often include both experimental and observational data in a meta-analysis, but I will code that. So the great thing about meta-analysis is anything that you think may explain variable results, like whether or not the data were collected through an experiment or observation, you can code that as a variable in your metadata set. And then you can formally test, are the outcomes different based on whether it was observed from an experiment or an observation? And that's a really useful test, right? It can tell us that, well, when it's done experimentally, they tend to observe this, but if you do it observationally, then we tend to see something else, maybe a smaller magnitude effect or even a null effect. So absolutely think combining experimental and observational in a single meta-analysis is great, but then we should probably test whether that's a source of heterogeneity.
Hi, Kasey. I have a question.
Sure.
So when is the scope of a meta-analysis too big? I don't know how to phrase that question properly, but yeah, when do you have to sort of evaluate that your scope and that it's not too big, and so your meta-analysis becomes just like an overwhelming amount?
I mean, I think that's a really easy thing to get tangled in a meta-analysis that's too big, and then you're on it for so long. And you're including, at that point, so much literature that there's going to be tons of significant sources of heterogeneity that aren't necessarily all that informative. So I think, they may be obvious. I personally like a meta-analysis that's rather focused on something that's a real problem. Like it's a question that everybody is excited about and interested in, but it seems like we just don't know. Why is it sometimes yes? Why is it sometimes no? And we can rein it into like a body of literature that tests that and has this lack of consensus. And that's where meta-analysis is most helpful to identify those sources of heterogeneity. I think if you get too broad, it's first of all, just too challenging. And second of all, the sources of heterogeneity can become confounded with each other, and you can end up with a really obvious answer like overall yes. So that's only like sort of helpful, but I guess I think in the end, to me, a more focused meta-analysis is actually the most helpful.
Thank you.
Yeah. So do we have separate review protocols? Absolutely not. If we're collaborating on one review, it's essential that we all have participated in developing this review protocol together. I mean, that's actually one of the biggest sources of inconsistency in a review, is if different people working on the same review are assessing literature according to different inclusion criteria. Because now we've introduced heterogeneity, which is the reviewer identity. So actually, we do track that when I do meta-analyses with multiple people extracting data. We include our names as a potential random source of heterogeneity in case there happens to be some like systematic inconsistency among us. And we test that, and we hope that it's not significant, because it shouldn't be. We spend a lot of time in the beginning training each other and trying to really capture consistent approaches. I think that's essential. In some ways, it's easier to do a review or a meta-analysis on your own. But on the other hand, then you don't have somebody kind of checking you. So I think collaborative reviews and meta-analyses are really great, but you have to be really careful setting it up. Meta-analysis, if there's variance, if hardly ever reported. Well, the problem is the definition of a meta-analysis is that we're statistically analyzing pooled data that incorporates between and also within study variance. So if studies don't report variance, they actually cannot be statistically incorporated into the models. They would just be excluded as data points. So if a large part of your literature doesn't report variance, it's not suitable for meta-analysis, because you just technically can't do it. And I think if what you're doing is you compile that literature and then you're like, oh, shoot, like 70% of my data points are going to be excluded when I run these models, then you probably want to pivot and do a qualitative assessment, right? Because that particular field of group of data are not conducive to meta-analysis, which does require within study variance. That's the whole point of it. And sometimes this happens, and sometimes you can do like a combo between a qualitative, quantitative. I've done this before where only some of our data were conducive to meta-analysis. So we did like a mini meta-analysis just to kind of see what was going on, but we couched that within a qualitative review that encompassed all the literature. Good question. All right. Well, you can keep asking questions in the chat, but I think we'll just carry on. Now that we've gotten through this first part of starting a review, which is the hardest part. So don't worry, that's a lot of steps and a lot of effort, but it will hopefully set you up in a really strong way. So now you feel confident and you're ready to approach the actual review. The first thing, of course, is going to be like formal literature searches. So in these first steps, you've probably also gained a good familiarity with the literature. You've read a lot already, but now you want to do systematic searches so that you're confident you're capturing all the literature on the topic that fit your inclusion criteria, so that when you run your assessment, whether it's qualitative or quantitative, you feel like you are addressing all the evidence that exists, right? So basically, searching the literature has also a lot of steps, but these are easier. So we're going to get through this a little bit faster. Defining your search terms is basically coming up with the string of terms that you're going to enter into Web of Science. Boolean search terms are the most common, and I think the most robust, because they can help us do all of the relevant search terms in one string. So I would say that what you want to do is take some time to practice in your search system with different kinds of search terms to see that it's catching the papers you want it to. And I would refer to this as like a pilot. This is pilot searching, right? Once you feel confident in the search terms that you want to use, then you develop your official search term list. And that is reported in your review. So this is like an important part of your methods, right? So this is actually copied straight out of one of my meta-analyses. So we did concatenated search terms, so Boolean searches in Web of Science core collection, calling these years. And the terms were leaf age or development or stage, phenology. And so it had to include this, one of these kinds of words that had to do with plant development. And then it also had to include plant defense or resistance, secondary, all these terms relating to plant defenses. And you can see I did things like question marks because defense can be spelled with an S or C, and I want to capture both of those. Chemistry has this asterisk after I because it could be chemical, chemicals, chemistry, chemotype. Oh, that wouldn't have been caught there. Same for herbivore, herbivore, herbivores, herbivory. So all of these then is my like official search terms that I run the meta-analysis on, and I have this recorded, and all the papers that it returns are my search returns. So to get to this stage, I did a lot of practice first, which is totally valid. You want to do that so that when you run the search, you feel confident it's getting what you want it to get. Now, what search systems are you going to use? This is actually not a super easy decision because there's a ton of variability in the reliability and precision across different search systems. So I'm going to refer you to this fantastic reference, this paper here. They did this really great systematic trial of 28 different search systems, and they had many different criteria, but basically they were looking at precision, recall, and reproducibility, right? These are things that resonate with us as scientists as part of the scientific method. And then they have really great tables documenting the sort of whether all these different search systems met these criteria. And some key takeaways from this, I refer you to this to go get more information to decide what system you want to use. But some key findings are that they found that Google Scholar is inappropriate as a principal search system de facto. And that's because it has low precision. They don't handle Boolean searches or parentheses. So that's part of how they have low precision, but it also means they have low reproducibility. So Google Scholar could be used as a supplementary search system, but should not be primary. Web of Science is robust and is a definite principal search system that's recommended. That's the one that I primarily use. Well, that's the one I always use. But one interesting recommendation is that there's different coverage across different search systems. So depending on your topic, Web of Science probably doesn't catch all the journals of interest. So they recommend using at least 2, maybe more different search systems for any meta-analysis or systematic review. I typically in ecology and evolution see Web of Science and Google Scholar being used. I find Google Scholar to be really unreliable, so I don't use it. And then that gets me thinking about what are the other ones in here, looking at their tables that are going to be helpful, and different ones for different meta-analyses in my experience. So this is a difficult decision, but it will affect the literature that you compile. So you need to be careful about it, and then you need to report it is the main thing. After you have your search terms and your search systems picked out, now you run your searches, right? So now you just do your official search. When you run your official search, you want to record the exact terms that you used, the systems that you used, and those details. So for Web of Science, you would want to report that it's the core collection and the years of coverage. That's an important detail. In Web of Science, you can always bulk download all of those search returns, including the title and abstract and everything. And I always do that right when I run the search, because that's then the data that I'm going to be assessing in my first assessments. You also want to record the date that you ran the search. And this is important because obviously, if it takes you a long time to do your review or meta-analysis, in the time that passes between when you ran the search and when you publish, more papers have come out. So you need to be transparent about, like, I ran the search in November 2022 so that people know, okay, anything published after that wasn't included. I'll come back to this point later, but writing the date is really helpful. And then here's where you would need to consider and take notes about other types of searches you do, gray literature, theses, etc. And there are good recommendations of database or search systems to help identify those. Always one of the key points is to take good notes so that there's a lot of transparency about how you compiled this data, because this is your data collection, right? This is how you're compiling the data that you're going to analyze. Once you get all those returns that you get from your running your searches, there's an initial assessment. And the initial assessment I include in a data sheet along with all that bulk download. So the same data sheet that includes all the bibliographic information and the abstracts for those search returns. I then just add some columns that are like, you know, abstract assessment and does it pass or fail. So in this initial assessment, I recommend just reviewing the titles and the abstracts. Often reviewing the titles and abstracts will help you make a first call about whether a paper is at least not meeting your inclusion criteria. It may not confirm that the paper fits all your inclusion criteria, but it helps you identify some no's, usually a lot of no's. So you have to have your inclusion criteria handy here, of course, for these assessments. And also for any paper that you are going to reject at this stage, you want to record why. Right? So here's our consistency and transparency really coming out. We don't just exclude papers for no reason. We need to have some reason for not including them in our assessment. So I have a column of the reason that I excluded them. And I always base it on my inclusion criteria. That's the whole point of making inclusion criteria. So you have reasons for not including a paper. For all the papers that then pass this initial assessment, I get the PDF so I can do the full assessment. And at the end of this initial assessment, you want to have pass-fail data that you can report, right? So you have the total number of search returns, and then you have the percentages of those that pass the initial assessment. This is just showing you what this data sheet might look like. Here are the authors. So this is just what Web of Science will give me: the authors, the publication year, the journal title, the volume pages. The abstract would be here, but of course I didn't include it in this, but I would read the abstract here in this data sheet. And then if it didn't pass, I have a reason why. So this is for a meta-analysis on coastal plant salinity tolerance. And I have different reasons for why a paper wouldn't be included in the meta-analysis. And then I have papers that, as far as I can tell from the abstract, seem to fit all my inclusion criteria. I'm giving all those yeses at this point. I don't know for sure yet because I haven't looked at the whole paper, but they passed my initial assessment. If it turns out you have a low pass rate, do you consider altering your scope or question and redo it? Yeah, I would say if you have, probably the main issue that I found if I get a lot of rejections is my searches weren't very good. So my search terms were catching too many papers that weren't related to my topic. So they're too broad. So that's an easy thing to fix, to go back and pilot search terms better. If you actually think that your scope is not producing a substantial enough literature for a proper review, then hopefully you caught that earlier. If not, you could still do like a mini review, but it might be more interesting if you broadened your scope to a bigger question. But yeah, I would say if you're ending up with few papers, I mean, it could be the search terms or the search system. That's the other thing to consider. Is the search system you're using not catching the journals or the places where you think these studies are? All right, now for those ones that pass that initial assessment, they get the full assessment, right? And so the full assessment is looking at the whole paper. So here's where some of those inclusion criteria I couldn't actually address initially, like the quality of the experiment and the availability of data. So at this stage, I do read the whole paper, but after you've read a certain number, the intro is usually pretty similar, or it depends on what the scope of your study is. I do read the whole paper, but I pay really careful attention to study design and data, because those are usually the deal breakers. If it's not a robust study design or there are no data, and for the data, it's especially for a meta-analysis, then I can't include them. So then they, at this stage, get a no. For anything, and I would record why that is. For anything that passes, these are the ones I'm holding to be actually included in the review or the meta-analysis. And sometimes as I'm doing this full assessment, I'm actually reading them as as if they're ready to go into the meta-analysis or review. So I'm taking all my notes and everything at this stage. Sometimes you're able to do this quickly and you can come back to a more in-depth assessment, but I find it more efficient to just, while I'm doing the full assessment, I'm actually reading it for content like I'm reviewing it. And then finally, taking care with the numbers so you can report the statistics again of the total that actually make it through both of these assessments for the total number of studies that are being included in your review. And I think that Anthony's point is good. Sometimes I've seen that the inclusion criteria is so strict that the number of papers that pass through the full assessment is very small. And to me, they're missing a lot of relevant literature. So I feel like this is definitely something that can happen. And I could assess why that was because they reported their inclusion criteria. And that's the main thing, is whether the reader can understand why the ending number of papers is so few or relatively a lot. That transparency is really the key. It's of course, always the author's prerogative to be as strict or narrow as they want with their inclusion criteria. But as a reader, then I can understand what's going on. So in the full assessment, I use data sheets. And this is because in the medical and sociological fields, they use data sheets. This is data collection, especially if you're doing a meta-analysis. So as I'm reading those papers for the formal assessment, I'm looking for specific information that I know I'm going to want to populate my metadata set with. These are the factors that I think may be driving heterogeneity across studies. These are the factors I'm going to test with statistical tests as potential explanatory variables of the data. So I'm just showing you again for this, using as an example, this current meta-analysis I'm doing on salinity tolerance. And my data sheets, this is actually what my data sheet looks like. These are the things I record when I read a paper. This is the information I'm pulling from those papers and recording so that then I can populate this into my data sheet, my data set. This is what all these data, and really the ones at the bottom are about specific data that I'm going to extract for the meta-analysis. So here's one of these now filled in with the information from a paper. And I'm only showing you this to emphasize down here, I actually take really careful notes about which data I'm extracting for the meta-analysis. And that's because often a single publication can include many different types of tests that you want to include in your meta-analysis. And you want a record of that, because each test that you extract from a publication is an independent data point in your metadata set. But if you're extracting multiple test statistics from a single publication, they're not independent of each other because they're related by that same publication. So we want to account for that by including publication as a random factor in our model. Anyways, the point here is I have a record of everything, right? This makes it really easy for me to then make my metadata set, because as I'm doing that formal assessment, I'm pulling this information that I want. If I'm not doing a meta-analysis, I still use data sheets because I try to be really systematic and consistent with how I do this. But my data sheet wouldn't include that bottom component of the actual where the data I'm extracting. It includes the notes from when I read the paper that I think are important, that I know I'm going to come back to in my discussion in the review paper. So I really like integrative reviews too. Qualitative reviews, I've done more of those than meta-analyses actually. And I try to be systematic in the same way, but then of course it's more about interpretation. So these notes down here really help me sort of think about how it's all coming together. Okay, so to sum up this assessment stage, whatever you're going to do with the evidence, qualitative or quantitative, this is our data collection phase, right? This is when we're compiling and assessing the data that we mean to then assess in the review. So, but emphasizing this a lot, having detailed notes is so helpful. That helps you create a record of what you're doing and the decisions you're making. Then when you include that in your paper, there's transparency to the reader. Having the protocol on those data sheets really helps with consistency. I know we all have our own biases. And so I try to account for my own personal bias by using these criterion data sheets so that I'm handling each paper consistently and fairly. And I think that really is important if we're trying to look for general patterns. And then finally, all of this should be reported in your paper, these report statistics. How many search returns did you get? How many of those were rejected? And how many of those passed to inclusion? And this helps the reader understand your methods and how many studies ultimately are being included in your assessments. So this is what Yinfan was asking about. What if you discover a whole set of related topics that should be included in your review, like as you're going? And as I mentioned, when she asked that question, you can always stop and do additional searches. Of course you can. Like, this is sometimes inevitable because, especially if you're new to a field, you discover that you missed a way that people are referring to this phenomenon, and you discover it while you're doing your assessment. So running additional searches is fine. Again, you just want to make records of all of this. So you want to, again, include those search terms, the search details, and the date you ran it, so that if you've done multiple searches, you have dates associated with that. And if you do all of this, then it's fine. You're doing the same systematic approaches. You're just doing it multiple times. If you don't do this, then you run the risk of mission creep, because now your inclusion criteria and your scope is changing midstream when you weren't careful enough to make sure you got all the relevant evidence. So that's the thing you have to be careful about if you're sort of pivoting mid-process. At the end of all this, there's actually usually a couple final searches that you need to run. So this is why running additional searches isn't problematic. It's just part of the process, and you just take careful notes about it. I usually have some really specific additional searches I do right at the end, like after I've compiled all of my evidence assessment, I do some final searches. And the reason for these final searches is, first of all, reverse and forward searches are super helpful. So this is actually can be ongoing. But as I'm reading papers in that formal second assessment, I always look at the bibliographies to find other papers that sound relevant, and then I add those and check them too in a formal assessment. So this is the reverse, or actually this is, yeah, reverse searching is looking at the papers cited in the papers that meet my inclusion criteria. And then for really good papers, reviews, or some of the most exciting examples, I do forward searches. I look at all the papers that have cited that paper, which you can do easily in Web of Science. And this reverse and forward searches are actually a really productive way to identify additional relevant papers. I'd say probably half of the papers that end up in my assessment come about through these reverse and forward searches. So don't skip these. They're really helpful. And then finally, depending on how long it's taken me from when I did the initial searches to when I'm actually done compiling the literature, I usually do some final catch-up searches just to fill in any papers that have come out since I ran the search. So if I did a search in 2020 and I'm right now, I have my data set, I'm ready to do the final steps, I would do a quick search to catch everything that's come out in the last 2 years. So you can constrain Web of Science to just searching these 2 years, and it will just report to you anything that you may have missed because it's come out since you've been doing all this. And then, of course, you always want to report everything. So this is just reminding you again to, as you do these additional searches, keep track of them and report them ultimately as part of your total statistics of how many papers you assessed and included. And then at this stage, you probably need to update your review protocol to reflect all of these additional method steps of actually running the searches and compiling the literature. And at this stage, we hopefully have our body of evidence, right? We've done all this work to identify the group of studies that include the information that we want to review. And so the final bit is going to be about how do we actually do the review, qualitative or quantitative. And this is where we're going to go into a short primer on how to actually do meta-analysis. But we'll also talk briefly about qualitative assessment. So this would be a good point too, if there are any questions about searching the literature or anything we've talked about. Or we just keep going. All right, let's do it. So now that we have our body of literature that we want to review, what comes next? So here we are. At this point, I've shown you how to do systematic searches, right? So we've basically moved away from considering these, which have a real value, but not what we're talking about, to thinking about how do you want to assess the literature? Are you doing it with a narrative review, which to me, these are all the same thing. It's just different ways that people talk about these narrative reviews that are based on systematic searches, or are we going to do a meta-analysis? And I would say qualitative reviews are great. These are especially ideal if you're talking a lot of theory or you're synthesizing across fields or approaches that are too different in the types of treatments or data that they include that you can't combine them into a single meta-analysis. So it's like the apples and oranges problem. If you have that, then a qualitative review might be the right approach. The thing about qualitative reviews is it can be prone to bias if you're subjective in how you assess the studies. So, you know, just being aware of that, I think helps to some degree. There's always going to be some subjectivity that we can't avoid, but trying to be general is good, or objective. I'd say one thing that often happens with qualitative reviews is people are very tempted to do vote counting. And vote counting is a really misleading and not robust approach. So if you're going with a qualitative review, try to avoid the temptation of vote counting. And I'll come back to that in just a second. Oh dear, look, this should be quantitative reviews. Quantitative reviews like meta-analysis are especially useful when you have a lot of studies that have similar approaches and outcomes, and that means treatments and data, and when there's no consensus, like I mentioned before. The con of a meta-analysis is its additional work because you have to extract all the data from those studies instead of just reading those papers. And it can, I think, lead to a false sense of objectivity or generality. I think people have the sense that meta-analyses are the most robust, but it's always the case that the quality of a meta-analysis is going to depend on the quality of the data and the scope of those data. So it's not necessarily more general or more objective. You need to then assess those search methods and also follow up with assessment of publication and other types of bias. So meta-analysis can be really robust, but not necessarily. So you have to just be careful of falling into that assumption. All right, so what is vote counting? Vote counting is simply when we count up the number of studies that have significant results for or against a particular prediction. So we're sort of trying to do a quantitative assessment because we're counting, we're producing a number, but it's based on study significance, which we know can be terribly misleading and not really a measure of the magnitude or the robustness of a study effect size. So one example would be, say in your review, you want to say something like 55% of studies reported that drought significantly reduced plant survival. It's the 55% that's vote counting. It means you counted up across all the studies, how many of them reported a negative significant effect. Vote counting is frowned upon, like I said, because it's based on significance. And it's sort of trying to be, it's posing as quantitative when it's actually not. So try not to do that. If you really want to do a quantitative assessment of a body of literature, you should do a proper meta-analysis. And the meta-analysis has the benefit of weighting effect sizes by the within study variance. That's the real key of meta-analysis. If you're just basing it on statistical significance, then you're not accounting for that variance, and that's problematic. So don't do vote counting. If you're going to do a qualitative assessment, just discuss the literature and maybe trends and things like that, but don't report these quantitative statistics. And then for the rest of the time, I'm going to talk about meta-analysis. So meta-analysis is great. It's a really wonderful approach and set of tools to synthesize the data from the studies. So for that body of literature that you identified in those systematic review steps, in a meta-analysis, we're actually extracting the data from those studies so that we can analyze all of the data across the body of literature in some statistical models. That's what meta-analysis is. And it uses effect sizes, which is a test statistic from those individual studies that is similar across studies. And what kind of effect size you use depends on the study. But the key thing is that meta-analysis includes both within study variants, that's really important, but also between study variants. So we can look at sources of heterogeneity. We can identify variables that statistically explain variability across the studies. And that's really helpful when there's a lack of consensus. All right, so how do we extract the data and run these analyses? Well, I will point you again to this wonderful handbook, which is super helpful and goes over a lot of these mechanics of doing meta-analysis. I think it's a really great book, but there's lots of wonderful resources. So I'm listing just a few people down at the bottom here who are really offering great information over YouTube or software or publications on different aspects of meta-analysis for ecology and evolution. And I find their work to be especially helpful and applicable to ecology and evolution. All right, so how do we know what effect size to use? Well, that depends on your question and the kinds of studies that you're testing. The ones that I have the most experience with are group comparisons. So these are usually experiments that are testing some treatment effect compared to a control group. And the difference between those groups is captured by the effect sizes Hedges D or the log response ratio. Both of those are really useful effect sizes for reflecting group differences. There are some benefits to them. Hedges D corrects for a small sample size bias, which log response ratio does not, unless you do some other things yourself. But Marc Lejeune has a really nice paper that includes a correction for that in log response ratio. I've used both Hedges D and log response ratio. I think they're both really good. Log response ratio has the benefit of being more easily interpreted. So to me, it's a little more intuitive, but I think they're both pretty commonly used. Correlations can also be used or compiled through meta-analysis, in which case the effect size is Pearson's R. And these are just two of many, many, many effect sizes. So again, if you go to that handbook, they have 2 chapters, one on commonly used effect sizes and a whole chapter on not commonly used effect sizes for particular kinds of scenarios in ecology and evolution. So I definitely recommend that if you're stumped about what kind of effect size your meta-analysis needs, look at this book. They have great discussions for choosing the right effect size. Like I said, I usually do group comparisons like this. So damage would be like the treatment group, this is the control group, or a correlation scenario like this. The actual data that we have to extract to include in our metadata that's then analyzed with the meta-analysis includes the group means. So the means, as I'm showing you here in this bar, the standard deviations of those groups and their sample sizes. We need all of these components in order to incorporate that within study variance. That's really important because that's a weighting factor in the meta-analysis. If you don't have standard deviation sample sizes, you can't include it in your meta-analysis. For correlation, the R is the effect size, but you need the sample size. The weighting factor in a correlation is the sample size. So again, you need both of these in order to include this kind of study in a meta-analysis that's using a correlation effect size. Okay. Now, obviously, if these data are reported as just an archived data set where you can calculate those group means and standard deviation and sample size yourself, that's super easy and great and pretty common these days. If these data are reported as means and standard deviation or standard error, which you can easily convert into, and sample sizes in a table. That's also nice because it's super easy. If it's reported in a graph like this, we can still get those data. It's just tedious. So for that, we need to digitize this image of the figure, and then we can extract the estimates using WebPlot Digitizer. There's actually a whole number of tools that can do this kind of digital data extraction. I really like WebPlot Digitizer, and about a year ago, there was a poll on Twitter of people who do meta-analysis in ecology and evolution, and WebPlot Digitizer emerged as the most popular and most reliable. So I think it's pretty commonly used. If the data aren't reported, or the variance isn't reported, or the sample size isn't reported, here's where you either just exclude that study altogether, or you consider contacting the author. That's sort of your call as the scientist here. I'm going to show you just briefly how to use WebPlot Digitizer. It's this free online software, so it's super easy, and they have nice instructions on the website, so you can just jump in there and probably figure this all out. But essentially what's happening is you take an image of the figure from the paper. So I just copied this out of a publication and turned it into an image, and I imported the image to WebPlot Digitizer. And then basically I wrote the steps down over here in brief. So the program is going to prompt you to calibrate the graph. So for a bar graph like this, you calibrate only the y-axis. So you would put a point here for 0 and a point here for 20. And then once it's calibrated, then you click on the points that are the data you want. So on the mean, then on the error bar, and the mean and the error bar. And then you can come over here and view the data and download it as a CSV. And obviously the error bar is going to report to you like something like 18. So you just take the error bar minus the mean, and that gives you the height of the standard error, right? And there's your data. So it's not hard. It is a little tedious, but this is what we do. And then ultimately, all this extracted data goes into a database, right? So this is a meta database from a meta-analysis that I published in 2016. So I'm including reference because I'm going to incorporate that as a random variable to account for multiple effect sizes that I extract from a single paper. And then all these variables are things that I think might matter in terms of variability across the studies. So species and the family, what growth form the plant has, where that plant grows, where its biome is, the type of trait. This was on an induction of physical traits, the name of the trait. And then here are the data, the control group sample size, control group mean, control group standard deviation, and then the treatment data would have been the columns after that. So a couple things, including reference is important because that's a random factor that you should account for. Including species is important because then you can actually incorporate phylogenetics as a random covariate, and that's typical these days, and I think often helpful to account for non-independence due to phylogenetic relatedness among your species. And then these other factors are the ones from my data sheets that I thought could be explaining significant heterogeneity. So these are the ones I'm testing with heterogeneity tests. So once you have that metadata set, basically the easiest thing to do is use Metaphor in R, which is a package developed by Wiegtbauer, and it's a really great, easy to use package. And within Metaphor, you can calculate from those raw data, the means, standard deviation, and sample sizes. You can calculate the test statistic, log response ratio, or Hedges D, and it will calculate the within study variance. And then you want to use random effects models because that incorporates the within study variance, but also the between study variance that you need to do heterogeneity tests. If you run a random effects model without any modifiers, without any explanatory variables, you're just going to get the overall pooled effect. So that's across all the studies, the overall pooled difference between, say, the treatment and the control group. That may be useful for answering the question of, is there a general pattern in the data? But probably more useful are heterogeneity tests, right? So these are the sources of heterogeneity within the data set. These are your predictor variables, those things that I thought might matter, like growth form or trait type or biome. Again, you want to use a random effects models, but with modifiers. And then from all of this, you get test statistics and degrees of freedoms, everything like you would expect. And they give you group estimates that have 95% confidence intervals associated with them. So to then sort of display these group pooled effects, you might do something like this. So this was from a meta-analysis looking at how traits change as leaves mature and develop. And I looked at nutrient content, secondary chemistry, physical traits like trichomes and prickles, and indirect defense traits like nectar and food bodies. And what I'm showing you here are the estimated group log response ratios and 95% confidence intervals, but I back transformed them into percent differences between young and mature leaves, because to me that's easier to understand. So that's the benefit of log response ratios, is you can do these back transformations to get percent differences between your groups. In this case, the groups are young, immature, and then mature leaves. So for nutrients and secondary chemistry, there's a significant decrease as leaves mature. So nutrients become more diluted and secondary chemicals become more diluted. And I know that's significant because the 95% confidence intervals do not overlap with 0. And then this is the number of effect sizes that went into that group estimate. So the number of data points from different studies. On the other hand, physical and indirect traits increase as the leaf matures, but these are smaller number of replicates, and you can see the 95 confidence intervals are much greater, but still statistically significant. So there's no comparable back transformation for Hedges D. That's why I think Hedges D is just less intuitive than log response ratio. All right, so now you've done your meta-analysis, you have these heterogeneity tests and maybe the overall pooled effect, and you even can show it in these kinds of graphs. Now, how do we sort of know how far we can generalize these results? Here's where, again, you need to consider any potential bias in your review, your meta-analysis or your qualitative review. So I think you need to reconsider here your inclusion criteria, because in your discussion or in your review, you need to be always couching your interpretation within those constraints. Sometimes if your scope is very narrow, then you can't really generalize very far. And if you're going with that approach, I think you need to be really careful to justify why you're having such a narrow approach early on, because otherwise I think we could interpret this as being so narrow that it's not really that helpful. It doesn't generalize beyond just that narrow scope. If you've tested a whole bunch of sources of heterogeneity and they're all statistically significant, that can be really informative, but it may be less informative if those different factors are confounded with each other. So you should detect or investigate through tests of independence your different sources of heterogeneity. So one example is, let's say in your data set, maybe all the herbs and forbs happen to be sampled in temperate systems, while the woody species happen to be sampled from tropical and boreal sites. So now you're going to detect a significant difference with respect to growth form, herbs versus woody, and also a significant effect of biome, temperate versus tropical and boreal. But you don't really know what's going on, right? Because they're confounded. So using tests of independence, you can document whether your heterogeneity factors are confounded so that we can more precisely interpret what's going on, right? In this case, we wouldn't know what's going on, but we want to acknowledge that. And then finally, how much are your results driven by publication bias? Say the most common form is that null or negative results may be published at a lower frequency, and that may overestimate the true effect of a particular treatment or factor. So we can test this formally using funnel plot analyses, and funnel plot analyses look like this. So basically all we're doing is investigating the relationship between the mean and the sample size. So the mean meaning the effect size. So in here, the effect size is on the x-axis and the sample size is on the y-axis. This line is the pooled mean of your whole study. and the dots are each independent effect size. So what we expect is that when there's small sample sizes, there's a lot of variability in effect size, right? They're spread out all over the place around the pooled overall mean. But for studies that have large sample sizes, they sort of correspond to and align very closely with the overall mean of the whole data set. That makes a lot of sense. So this is what a funnel plot looks like in a really balanced scenario where there's no evidence of publication bias. Lots of spread down here, and they coalesce up here on the actual mean. Here's what it can look like if there's publication bias. So again, the same thing. This is the effect size. Here's my overall effect size. You can see it's not 0, it's something over here, 0.3 maybe. And I'm using standard error as an estimate of sample size. A small standard error reflects a very large sample size, and a large standard error reflects a small sample size. So what we have is actually quite a lot of studies up here, and then one outlier that had a really small sample size. But also what we're seeing is that there's more studies over here outside of that funnel than on this side, and that indicates a positive publication bias for studies towards a positive effect size. And relatively fewer studies are getting published that have a null, a 0, or a negative effect size. So if we look at the actual correlation statistic between effect size and standard error, it's significant in this case. Oops, this is hard to see. It didn't show up quite right. It's P equals 0.0145. So that indicates that there's a skewed overemphasis, a publication bias for positive results. There's nothing we can do about this, but we want to know if this is happening because it could mean that there are relatively some studies out there with negative and null results, and we're probably overestimating the overall true effect size of this particular treatment. Okay, and then you might also want to look at other potential data gaps in your meta-analysis and report these. So you may find that some groups have really low replication. So those are areas like maybe biomes or trait types or species that are underrepresented in the data set. It's often that they're understudied taxa. So for example, there may be an overemphasis on model species or crops. There may be understudied traits, reflecting an overemphasis on easily measured traits, which swamp your analysis. Other forms of data are often lacking indigenous biocultural knowledge that mean we don't totally know what's going on with our particular question. I think biogeographic data gaps are really important, and it's not because those data don't exist, it's because they're often not as accessible to people working in the global north in meta-analysis. So we're missing data from Africa and the global south often. And this is a really important point because people often talk about global meta-analyses, but you need to really look at the biogeographical scope of your data set. And if you don't have strong representation from Africa and the global south, for example, it's not a global meta-analysis. I think often there are data gaps from local scientists. So there's an over-reliance on parachute science in many cases, and that also then leads to an overemphasis on easily measured traits. And finally, there may be different tools or approaches that aren't very well represented. That can be an important and exciting opportunity for future directions. Maybe it's because there are new tools or new approaches, or something of concern, maybe there's not equitable access to some of these tools and approaches, and that could be confounded with, say, biogeography. All of these are just helping us acknowledge the constraints of our meta-analysis or review, right? So it's not that these are things that are necessarily deal breakers. It's just to help us be careful about how far we try to go in generalizing from our results. Our review is constrained by the evidence we're assessing. And if there are these kinds of data gaps, then we want to be aware of that and we want to discuss it. In some ways, this is a great opportunity for highlighting future research directions or ways of trying to increase equity in science. So these are just really important talking points. If you notice gaps in the geography, it's not too late. Of course, you can always backtrack. One of the real issues is language barriers, which I mentioned before. So sometimes to capture some of these underrepresented biogeographical areas, you need to broaden the language scope of your meta-analysis. And here's where getting a collaborator can be really productive. So I'm seeing this more and more of people getting really international meta-analysis teams so that they can capture a more representative and greater diversity of research. And that brings us to the final point, which are resources. So, right, the resources is that list of publications that I shared with you in the chat at the beginning. That is just a partial list of some papers that you might find useful as you get going on this yourself. There's tons of great resources out there. So I really encourage you to get into that so that you can strengthen your own expertise in how to do this. And I think it will result in a much stronger review. So then my final take-home messages are just that writing reviews is really great. It's a great contribution to a field. It's great personally because you really become an expert on a topic. And that's really exciting and satisfying. I think it is hard, but it's so much easier if you sort of have this kind of strategy to help formalize the process. To me, it's much harder to do a haphazard review because I'm all the time feeling like I'm missing information or I don't know exactly what my goal is. So spending the time to set up the systematic review protocol at the start results in so much more consistency and transparency and ultimately a much more robust review. And I think it also alleviates the stress of not knowing what you're doing as you go. And then adding a meta-analysis can be really helpful. It's not necessary for a good review to be a meta-analysis, but in some cases, it's so helpful to understand why there's so much heterogeneity in a body of literature. So I think that's it. I'm actually gonna just wrap this up here and put the Botany360 survey back into the chat. And also I'll put the link to the bibliography in there again. Here's my email address. If you would like to ask me any follow-up questions after today, I'm happy to discuss over email. And if you're thinking about writing a review and you'd like to discuss how to go about doing that at American Journal of Botany, that's where I serve as the reviews editor. I'm more than happy to discuss your ideas with you. We do take formal proposals on this reviews@botany.org email address, and you can also just email my Hawaii account.